welcome back to another science session, and today we are going to talk about nucleotide metabolism. Okay, so the starting point would be nucleotides are built from what? From a nitrogenous base, a purine or a pyrimidine. Wait, let me write that down. Where's my blackboard, my whiteboard, I mean. So it's made up of a few components. You have a nitrogenous base, which is a purine or a pyrimidine, a 5-carbon sugar, which could be a ribose or a deoxyribose, DR, and one or more phosphate groups. That's a nucleotide, okay? Meanwhile, a nucleoside is just the base and the sugar. So nucleoside, meanwhile, nucleotide has the phosphate. And yes, it's essential or important that you know how to recognize these structures. Science! Hello, Vince VA. Yes, we are talking about science. What is happening? Why is my... Oh, it's okay. We'll just move it. This is nucleotides. Yes, science. Now, you have purine degradation. So purines, like adenine and guanine, they are broken down through a stepwise process. After they get broken down, the ending is uric acid, okay? And the uric acid is then excreted in the urine. And it's important that you remember this pathway, because if you have defects or imbalances, that can lead to gout. And that is where like uric acid crystals accumulate in joints. So yes, again, purines such as adenine and guanine, purine, AG, broken down through a stepwise process in the form of uric acid. And if you have defects or imbalances in that, you get gout. And then you have salvage pathways, which is in addition to synthesizing the different molecules or nucleotides from scratch, the cells can recycle the bases. Wait a second, why isn't the stream buddies working? Let me try to type science. Will it work? There you go. It should work, right? It's okay. If it doesn't, it doesn't. So you have nucleotide metabolism. It's tightly controlled to maintain. Yeah, the stream buddies are not popping up. It should work. Refresh. I'm gonna refresh it a tiny bit. There you go. It now works. You see? There. Nice. It's good that the hats are also the mini emotes. And you can change your hat by typing exclamation point. Hat. That one will change the hat. And then when you oh nice. And then when you chat, it has the ... Okay. So nucleotides, feedback inhibition by end products such as ATP, GTP, and CTP would ensure that the nucleotide levels remain appropriate. And this regulation will help the cells respond efficiently to growth, division, and stress signals. Okay, good. So don't forget, this is where ATP, GTP, and CTP comes in. What is the next one? We have a couple of nitrogen heterocycles and nucleotide bases. So let's just scribble some of them here. Let's erase them first. Erase. Okay, it is now erased. So you have the cyclopentadiene, which looks like this. This is the Superman symbol. So that's the cyclopentadiene. Oh, the lipid stream earlier was so funny because more than 12 of the wiggly bobbleheads were happening all at the same time, so it was so funny. Okay, so cyclopentadiene, 5-membered ring with 2 double bonds, not aromatic, but it will just serve as a structural comparison. And then we have the pyrrole, which looks like this, like that. And then you have NH, that's a pyrrole, double R, 1 nitrogen. It's for heme, and it is also in purine synthesis. You have imidazole, another version of a 5-membered ring, and H, but you have a nitrogen over here, imidazole. So it is in the imidazole. That is in the side chain of histidine. And then you have histidine actually. So you have the imidazole. Nope, wrong shape. 5, 5, 5. We need it to be 5. Like this. Bam. Nice. Okay, you have NH over here. And then like this and like this. And then an N over here. You have the CH2. And then the middle C. And H2 and the COOH. That's histidine. And then you have heme, which will look weird because it looks like this. It has a lot more things going on. 5, yeah. It has a thing there, a thing here, a thing here, and it's all connected like that. Kind of like a something circle, but not exactly a circle. So that's a tetrapyrrole, a heme. Okay? And then you have different aromatic ring systems, which has more important relevance, like tryptophan. Okay? Tryptophan looks like this. Later on, we will actually have, like, when we are discussing the different proteins and amino acids again, I will try to explain the different side chains properly. But for now, the focus is on nucleotides. Wow, that is a horrendous hexagon. Again. Like this. Good. Nice. Okay. Now that looks like a hexagon. Like piatos. And then you have nitrogen. And H2. Nitrogen. This is a 5-member ring, so it's like here. Nice. Okay. So what's that? Adenine. Meanwhile, guanine would look like this. Another 6 and a 5 here. But here's the difference. Guanine has double bond O and H, double bond N, NH2. It looks weirder. And then this one is just N, and there, double bond. See? That's guanine. So the one that looks uglier is guanine. Prettier is adenine. I think. Just more stuff going on in guanine. Okay. So for adenine, that is a purine with an amino group at C6. It's found in ATP, DNA, and RNA repairs. And then guanine is also another purine and can form it has an amino group at carbon 2. And it forms 3 hydrogen bonds with who? Cytosine, which is where the genomic stability will happen. Okay, good. So just remember that pyrimidines have single 6-membered rings. Meanwhile, purines have a 6 and a 5 ring. So pyrimidines are uracil, thymine, and cytosine. Okay? We could actually draw them. Fine, I will. One moment. I will just put a sticky note. There you go. Over here. Okay, so uracil looks like this. All of them are 6-membered rings, so actually let's just start with writing it. Drawing it. Good. Okay, uracil, thymine, and cytosine. Uracil is double bond O and H, double bond, another NH, and this. So it looks a little bit symmetric in a way, but not exactly. Meanwhile, thymine, roughly the same starting point, except it has a methyl. So thymine has a methyl, uracil does not. Uracil is in where? RNA. Thymine is where? DNA. Okay, good. Next, cytosine. What's the difference? You have the same NH, double bond O, but here you only have an N because it has a double bond here, which has an NH2 over there, and then bam, a double bond. So uracil and thymine kinda look alike, but thymine has a methyl. The one that looks very different with an NH2 at the top and an extra double bond is cytosine. Alright, good. And we talked about the bases. The nucleoside of adenine, if you have the base, actually let's write that. This is a good one. It's a mini table. If you have a base, this is the base. This is the nucleoside. And then this is the nucleotide. Or nucleoside phosphate. Okay, good. So for the base, we have adenine, right? Adenine. This is adenosine. And then you have AMP, adenylic acid. And then you have guanine, guanidine, and then GMP, and then you have uracil, uridine, UMP, and then you have cytosine, which is cytidine, and you have CMP, bam. And then there are stuff called modified bases like xanthine and hypoxanthine. We'll just focus on the important ones. Alright, so how do we synthesize nucleotides? Because the stream title does say metabolism. Erase these first. Okay, good. Finished. So, you first have glucose. We start with glucose. And then you generate, at some point after a lot of steps, you make ribose 5-phosphate. And that is generated through the what pathway? Pentose phosphate pathway. Yes, it's actually important. So the key intermediates would be just something called 5-phosphoribosylribosyl 1-bisphosphate. Just remember, glucose to ribose 5-phosphate R5P. It will go through the thing using pentose phosphate pathway. Okay. And note that in pyrimidine synthesis, the ring is assembled before being attached to the PRPP. Meanwhile, in purine synthesis, purine, the one that has double, the purine ring is built stepwise. Alright, what does that mean? So let's erase this. Let's make it smaller so that the map can be expanded. So you have glucose to using the pentose phosphate pathway, and then you have ribose. Hello George Ross, welcome back. We are just trying to make nucleotides right now. It is called the road to nucleotide synthesis. That's where we are. And we're starting again with glucose, a thing that we know, sugar. And through the pentose phosphate pathway, you make ribose 5 bisphosphate, right? No, just phosphate. Ribose 5 phosphate. By the way, I've decided what the thumbnail for the Saturday powered stream. It's an Easter. Is it called an Easter celebration? No, it's just one of the Easter thumbnails that I was thinking about. But yeah, I'll use that. Okay, so you have ribose 5 this phosphate here. At some point, you convert that using ATP, right? And then you have AMP after. You have ribose 5 bisphosphate. Bisphosphate. If you want to make the pyrimidines, it becomes UMP, UTP, and then CTP. And then, if you want purines, their N is from glutamine. And then you get AMP and GMP. So that's the purine synthesis. This is purine, this is pyrimidine. It all happens from what? From glucose to ribose 5-phosphate into ribose 5-bisphosphate, and bam. So the first purine nucleotide formed is IMP. Okay. Remember again the PRPP 5-phosphoribosyl 1 pyrophosphate. Common precursor for what? For both purines and pyrimidines. Hurrah! Okay. Purine synthesis. Well, you could start. Let's see. There's a lot of stuff here, but we'll go through them slowly but surely. There you go. Okay. We start with... Who is this? Sorry, I'm zooming in. I can't see my text. Carbamyl phosphate synthetase. Okay, you have CPS2. It becomes something, and then dihydroxy, I think, acid, and then at some point you have UMP. So that's pyrimidine synthesis. Purine synthesis also ends with IMP. It will end that way. So just remember, the most important thing I think would just be the starting point. That they start from the same stuff. Now, if you want to break it down, oh, by the way, if you don't have this, if this isn't working properly in your body, you get gout. It's when uric acid will keep on like accumulating in your joints. So here you have AMP, right? AMP into adenine, and then you have hypoxanthine. It becomes xanthine. That's coming from GMP, guanosine, and then you have uric acid. Okay, so purine degradation leads to uric acid formation, and if you do not clear uric acids properly, you can get the gout. G-O-U-T, gout. And then you have a treatment for gout, of course. That's called allopurinol, and that will actually help because uric acid forms from purine degradation. And how allopurinol works, this is the spelling, allopurinol. It is a structural analog of hypoxanthine, and it's a non-competitive inhibitor of xanthine oxidase. So the inhibition would block both the hypoxanthine to xanthine and xanthine to uric acid. So that way your uric acid levels would decrease, and that would reduce the risk of crystal formation and joint inflammation. And this is also crucial. Yeah, this is crucial for controlling hyperuricemia in gout patients. Allopurinol. Okay, now you have purine salvage. It is a pathway that recycles purines back into nucleotides, and that would save the cellular energy, right? And if salvage enzymes are deficient, purines are degraded instead, and yeah, that's how we get the uric acid. Just remember, purine, if you have excess uric acid, aka excess purine degradation, gout-like symptoms. So there is a thing called that some diseases could be like the LESCH, L-E-S-C-H, LESCH, Nyhan syndrome. It's caused by HGPRT deficiency. You also have severe combined immunodeficiency. You have the von Gierk's disease, which is a deficiency in glucose 6-phosphatase. Essentially, you don't want a lot of uric acid in your body. Okay? Remember that one. Okay, so just mini questions. What's the difference between a nucleoside and a nucleotide? Nucleoside consists of the base, the nitrogen base, which is a purine or pyrimidine, and the 5-carbon sugar, which is a ribose or deoxyribose. While a nucleotide includes one or more phosphate groups attached to the 5-carbon position of the sugar. So adenosine is a nucleoside, and then AMP is a nucleotide, adenosine monophosphate. Again, nucleotide has the phosphate, nucleoside does not. How does purine synthesis differ from pyrimidine synthesis in terms of PRPP utilization? It's the ring. Purine rings are made step by step onto the PRPP. Meanwhile, pyrimidine forms the ring first and then attaches it. So again, purine step by step attaches it. Meanwhile, pyrimidine will form the ring first before putting it on PRPP. And then how is nucleotide synthesis regulated? It's regulated via feedback inhibition. AMP and GMP inhibit their own formation. If you have a lot of it already, you don't want to form more of it. Why do humans accumulate uric acid instead of degrading purines further like other mammals? That's because humans lack uricase, which converts uric acid into a more soluble allantoin, I think. And so that's why uric acid is the final oxidation product and should be excreted in the urine. And excess uric acid leads to the formation of gout. How does ribonucleotide reductase contribute to DNA synthesis? Well, ribonucleotide reductase would reduce ribonucleotides and supplies the building blocks for DNA. It uses thioredoxin as an electron donor, and it's tightly regulated. And note, it's for the S phase of the cell cycle, which I will talk about, I think, later if I haven't yet. Okay, how does allopurinol help gout? We talked about this. It is a hypoxanthine analog, and that is converted by xanthine oxidase to alloxanthine. And then it will inhibit the xanthine oxidase, which will prevent hypoxanthine and xanthine being made into uric acid, which will lower the body's uric acid levels. And then, what nucleotide metabolism defects cause immunodeficiency? So, that is the ADA. Wait, isn't that in one of the... What was the investigative New York show? Not CSI. The one that has an elevator music, the ADA, Casey Neistat or something. I don't remember. But anyway, adenosine deaminase deficiency leads to the SCID by accumulation of deoxyadenosine and DATP. And that can inhibit ribonucleotide reductase and kill your lymphocytes. So that's why it's immunodeficiency. And how do pyrimidine analogs interfere with DNA synthesis in cancer therapy? Well, drugs like 5-fluorouracil could inhibit the thiamidylate synthase, which will prevent the DUMP to DTMP, and that will block DNA synthesis. So again, drugs like 5-fluorouracil could inhibit something, and then after that it will prevent dump into DTMP, and which would block DNA synthesis. And then you have methotrexate, which will inhibit the dihydroxyfolate reductase, and that will reduce the supply for thymidine and purine synthesis. And just a mini note, if we want to design an experiment to test whether a purine salvage enzyme is functional in a patient sample, you gotta start by extracting lymphocytes first from a patient. Extract the lymphocytes, incubate them with a radio-labeled hypoxanthine. So you extract first the lymphocytes, and then incubate them with a radio-labeled hypoxanthine. After you have it radio-labeled, you use a thin layer chromatography or HPLC to detect the formation of the labeled IMP. And a functional HGPRT or purine salvage enzyme will be able to catalyze hypoxanthine to IMP, but if it is deficient, it will result in less IMP. So that's why we have to use a healthy donor as a control as well. So again, extract the lymphocytes and then incubate it with radio-labeled hypoxanthine, use TLC, thin layer chromatography, and then you can just see if you have the IMP or not. And then okay, you suspect that a compound inhibits de novo purine synthesis. How can I test it in cultured cells? First, you grow the cells in a medium with labeled glycine, and then track the labeled glycine track its incorporation into the nucleotides via mass spec. I like mass spec. If the compound will inhibit de novo synthesis, the labeled purines will be really reduced. And then if you do a rescue experiment and you add hypoxanthine, it's expected that you can restore growth if the salvage is intact. So again, grow cells in a medium with labeled glycine, and then track incorporation into nucleotides via liquid scintillation or mass spec. So yeah, grow the medium with labeled thing, track incorporation via mass spec. If the compound will inhibit the synthesis, then your labeled purines will be super tiny and low. And then if you do a rescue experiment by adding hypoxanthines, then you should be able to restore the growth. Alright, and then how would you determine whether uric acid accumulation is due to overproduction or undersecretion? You measure 24-hour urinary uric acid. So overproduction will show high urinary uric acid, about greater than 800 milligrams per day, but under excretion would show normal or low. So you have to combine them with different assays to test the rates. And then how could you test if a patient has the syndrome, the Lesch-Nyhan syndrome, due to HGPRT mutation? Sequence the HPRT1 gene from a patient DNA to identify the mutations, because if you sequence it, you might be able to see something. But alternatively, you can perform a functional enzyme assay with the patient's fibroblasts, and then label again their hypoxanthine. And then if you cannot convert it to IMP, then you know that they have a deficient HGPRT. Right? Right. And then just like a mini tidbit about purine purification. Well, protein purification, and first of all, why would we want to purify proteins? Well, it's essential because if you need to study the function of proteins, you need to make sure it's pure. And then you can also produce some therapeutics, like many biologic drugs such as insulin or antibodies require a very high purity protein for safety and efficacy, okay? You can also perform some structural studies like X-ray crystallography or cryo-EM or NMR. Those all require pure samples. And then you can develop some diagnostics, like purified proteins are used as reagents in ELISAs and other diagnostic tests. And you can also characterize some interactions, like binding assays, which are required to isolate the molecules. So the thing is, two things you must have before starting purification. You need the source and the assay. You need the source, know what biological material contains the protein that you want to test. That could be E. coli, yeast, or tissue. And then the assay, aka the method you use to detect or measure your protein, which could be activity assay, absorbance, antibody, tag detection. And then how does its flow go? I almost forgot that I had a whiteboard. I was so used to not having the whiteboard. Okay. Here, erase it. So again, you have when you do protein purification. No E. Purification. There you go. You need the source and the assay. And then the normal flow is you have the cell lysis, you break the cells open, and then you have the clarification or centrifugation. Centrifugation. And then you capture the protein. Then you purify the intermediate, polish, and then buffer. Okay, let me explain it one by one. So cell lysis is first you break open the cells, and then you centrifuge to filter or remove the cell debris. After that, you have to capture it. You have to enrich the protein from the lysate, maybe by affinity chromatography. And after that, when you purify, you can actually use ion exchange or something like a hydrophobic interaction chromatography. Then you polish it to reach high level purity, and then just do some concentration like dialysis or ultrafiltration. But then usually you just stop around here, the ultra polishing part. Okay, so now we have a couple of separation procedures. Let's write them here. Oops, no, this one. So this is how to separate stuff. Separation procedures, how to split them up. Okay, you have the characteristic, and then you have the actual procedure. So here you have the solubility. If the characteristic is for solubility, you can do salting in, salt in, or salt out. So for salting in, it will improve protein solubility at low salt by shielding the surface changes. Salting out, you use high salt to reduce the solubility. So again, salting in would improve protein solubility at low salt. I don't understand. I think that's flipped. Wait. Salting in improves. Okay, wait. Salting out uses high salt to reduce the solubility. That's confusing. Anyway, let's skip that. Okay, ion exchange chromatography. This one will hopefully make more sense. Ion, the ionic charge, that is the characteristic. You have ion exchange chromatography. Ion exchange chromatography. This one will separate the proteins based on net charge by binding them to a charged resin, and the proteins are eluted by increasing the salt or changing the pH. So separate them based on net charge by binding them to a charged resin, either cationic or anionic, and then you just increase the salt or change the pH so that the pure proteins are eluted. Then the thing that we really hopefully know by now is electrophoresis. Still ionic charge. Electrophoresis. So the proteins will migrate through a gel under an electric field. The speed is determined by size and charge. SDS page, the difference with SDS page and normal GE is that SDS page removes the charge differences by coating it, and then so the separation is just depending on the size, and it's primarily used to analyze purity and not exactly for large scale purification. And then you have isoelectric focusing, wherein the proteins are separated in a pH gradient gel until they reach the isoelectric point. If you have a thing for the characteristic of polarity, let me write that. For polarity, you have the absorption paper chromatography, reverse phase chromatography, hydrophobic interaction chromatography, and also, that one, yeah. All of these chromatographies just based on polarity between the molecules and the stationary phase. So for example, in the paper chromatography, it's less commonly used now for proteins, but it's very historically important for small molecule separation. Okay. Now we have the characteristic of molecular size, and that's where dialysis and ultrafiltration comes in, because dialysis, if you've heard of it, the dialysis, this one removes small solutes like salts using a semi-permeable membrane. And that's like the same thing, the dialysis that we know. We talked about molecular size like the gel electrophoresis. There's also the size exclusion. Based on size, large proteins elute first, small ones later, and this technique is very gentle and it preserves the native protein structure, and it's ideal for final polishing steps. Ultracentrifugation, just very high speeds, round and round proteins, and the proteins will sediment based on the size and density. It's used to separate very large complexes or organelles. While it's accurate, it's very time consuming, and it's more for analysis compared to very preparative purification. And then we have the binding specificity, which is affinity chromatography. This one is kind of expensive, but it's really great because it will be for specific binding, like histidine tags binding to nickel, antibodies binding to antigens, or substrates binding to enzymes. It's the most selective technique, especially when the tags are very engineered. But take note, it costs some money. Okay, so mini questions. Why is protein purification important? It's because some techniques need them to be pure. For example, purification would allow the researchers to study the protein's function, structure, and interactions in the disease. and it is important to create pure stuff for diagnostic reagents. What's the components required before initiating a purification strategy? You need the source and the assay, the source of your protein, the assay, aka the technique that you'll use. What distinguishes salting in and out? Salting in increases solubility at low salt. Salting out precipitates the protein at high salt. Oh, that makes sense. Salting out precipitates the proteins at high salt. How does ion exchange chromatography separate the protein? It's based on net charge. Ion exchange, because ion exchange. The proteins would bind to opposite charged resins, and then they're eluted if you increase the salt concentration or change the pH, which will alter the binding strength. And why is SDS page used in protein purification? That's because SDS page will denature the proteins and give them a uniform negative charge, allowing the just purely size-based separation. How does gel filtration chromatography work? It's known as size seclusion. Large proteins elute first, while smaller ones enter the pores in the resins and elute later. What makes affinity chromatography very, very unique and powerful? It's because it allows highly selective isolation of the target protein from a very complex structure. Okay? And what is the principle of hydrophobic interaction chromatography? The proteins would bind to the resins under high salt concentrations or conditions, and that's because of their exposed hydrophobic regions. You elute them by reducing the salt concentration. So as you can see, just play around with the salt concentration. Why are dialysis and ultrafiltration important even though they don't separate proteins? They're used for buffer exchange, salt removal, or protein concentration. Okay, I think for this mini part, wait, one more. Your protein of interest loses activity after salting out. How would you troubleshoot? Just change the salt concentration again to test it. But yes, so far, I think for both nucleotides and a little bit of protein purification, I think that's it for today, actually. But yes, thank you for coming here to watch the short stream, and that was it. Bye bye.